Good morning, everyone. Today, we are going to delve into reviewing past A-level examination papers. This session is designed to help you understand how to approach these papers effectively and ultimately enhance your exam performance. We will explore the structure of the papers, common question types, and strategies for answering them. Without further ado, let's get started. The eyepiece lens of a microscope can be fitted with an eyepiece graticle. Which of these statements about eyepiece graticles are correct? The answer is statement 2 only. Because the eyepiece graticle can help in drawing or estimating the proportion of cells relative to each other when used with a calibrated scale. Meanwhile, statement 1 and 3 is not accurate and not correct. For statement 1, the eyepiece graticle do not measure actual length directly. They need to be calibrated against a stage micrometer to provide measurement. Meanwhile, statement 3 considered is not correct because the eyepiece graticle itself does not change size. However, the field of view changes with different objective lenses, which means that the scale of a graticle needs to be recalibrated for different magnification. A student was asked to use the scale bar shown to calculate the magnifications of a cell on a photomicrograph. Which method would the student use to calculate the magnifications of the cells? The method that could be used by the student to calculate the magnification of the cells is have to measure the length of the scale bar in millimeters, convert to micrometers, and divide by 2. Which eyepiece and objective lens combination of a light microscope allow the greatest number of cells in the field of view to be seen? To see the most cells in a field of view, you need lower magnification. Therefore, the combination with the lowest total magnification is eyepiece multiplied 5 and objective multiplied by 10. This will show the greatest number of cells. Which role correctly matches each cell structures with its function? The answer will be microtubes form the spindle needed for the cell division, and meanwhile, rough endoplasmic reticulum helps in transporting protein. The answer will be C. Which animal cells would have the most extensive Golgi bodies? The correct answer is B because goblet cells are specialized for producing and secreting mucus. They have an extensive Golgi apparatus to process and package the mucus for secretion. And number 6, which of these processes will require ATP? The process that will require ATP is only semi-conservative replication. And for statement 1, transport of water in the xylem and facilitated diffusion of amino acid into the cells, they are not require ATP. So the answer is D, 2 only. Which concentrations could be produced by a serial dilutions of 8% glucose solution? So the correct answer is A because in a serial dilution, each step typically reduces the concentration by a consistent factor. For example, if each dilution reduces the concentration by half, starting from 8%, solution would produce the following concentration. 8 reduced to 4%, 4% reduced to 2%, 2% reduced to 1%, 1% reduced to 0.5%, and 0.5% reduced to 0.25%. A student wrote these statements about polysaccharides. Which of these statements are correct? Only statement 3 are correct because of in unbranched beta-glucose change, each monomer is rotated 180 degrees relative to its adjacent monomer. For statement 1 is incorrect because amylose is formed from condensation reactions between alpha glucose monomer, not beta glucose monomer. And statement 2 is incorrect because of branches in amylopectin molecules formed between carbon atom 1 and 6 on alpha glucose molecule, not as per stated in the statement, which is between atoms carbon atom 1 and 4. Number 9, which diagram correctly show the formation of a peptide bond between two amino acids? So the diagram that show the formation of peptide bond between two amino acids is A. Sugar with a ring structure can also have a linear structure, which sugar molecules could be presented by the linear structure shown in the diagram. This is alpha glucose only. Which molecules are globular protein? The correct answer is amylase and hemoglobin. Amylase, this is a globular protein which is act as an enzyme to break down starch. Hemoglobin, also a globular protein because it is responsible for transporting oxygen in the blood. Not DNA polymerase because this is a complex enzyme, but it is not typically classified as globular protein. Rather, it has a more complex structure that is not purely globular. As a result, amylase and hemoglobin are globular proteins. When hydrolyzed, which molecules have product containing a carboxylic group? The correct answer is phospholipid and protein because phospholipid and protein, when hydrolyzed, it produces molecules with carboxylic group. Meanwhile, polysaccharide do not produce carboxylic group when hydrolyzed. Lysosome contain many different hydrolytic enzymes that may act within cells or outside cells, and which process must occur in order for lysosomal enzyme to act outside the cells. To determine the process needed for lysosomal enzyme to act outside the cell, we have to consider how these enzymes are released from the lysosome. So the correct process is exocytosis. The diagram shows an enzyme is substrate and enzyme substrate complex, which statement explains how the substrate is able to enter the active sites of the enzyme. The answer will be contact between the substrate and the enzyme, which causes a change in the enzyme sheet. What could be used to calculate the rate of enzyme catalyzed reaction? To calculate the rate of an enzyme catalyzed reaction, you have to consider both. One is the appearance of the product, and secondly, the disappearance of the substrate. Cholesterol is an integral component of a cell surface membrane. Which statements about cholesterol is correct? The correct statement about cholesterol is it helps to regulate the fluidity of a cell surface membrane. Cholesterol stabilizes the fluidity of the cell membrane by preventing it from becoming too rigid or too fluid. Which type of bond in phospholipids has a role in increasing the fluidity of cell surface membrane? The type of bond in phospholipids that increases the fluidity of the cell surface membrane is B, because there the presence of double bond in the fatty acid chain create kinks, preventing the phospholipids from packing closely together and thus increasing membrane fluidity. Which of these substances can pass directly through cell's membrane without a carrier protein or a channel protein? The substances that can pass directly through the cell surface membrane without using a carrier protein or a channel protein are CO2 and C6H12O6, which can diffuse through the lipid bilayer, while Ca2 plus is typically require a carrier or channel protein due to its charge. Companion cells use ATP to move hydrogen ions out of the cells and co-transporter proteins to allow hydrogen ions to return with sucrose molecule. Which two processes are involved in this movement? The two processes that involved in this movement are active transport and diffusion, which can be explained. Active transport is used to move hydrogen ion, which is proton, out of the companion cells against their concentration gradient, which require ATP. The hydrogen ion then return into the cells along with the sucrose molecule through co-transporter proteins, which is a form of facilitated diffusion because it involves specific transport protein but does not require energy input directly. How many copies of each different DNA molecule will be found in a cell at the start of each of the stage of mitotic cell cycle? The answer will be D. This can be explained through G2 interphase. By the end of this phase, each chromosome has been replicated. So there are 2 copies of each DNA molecule. And during phosphase, the chromosome have condensed and are visible as 2 sister chromatid. So each chromosome still have 2 copies. And for the stage of cytokinesis, especially at the end of these processes, these cells have divided into 2 daughter cells, each with 2 copies of each DNA molecule. 3 parts of a chromosome and their functions are listed. And which combination is correct? The answer will be D, which is telomeres. The functions is to prevent loss of genes. The graph shows various distance measurement taken for metaphase of mitosis onwards. The graph are to scale when compared to one another, which will correctly identify the distance measurement for each group. The answer will be C. And 23, a culture of bacteria was allowed to reproduce using nucleotides containing the heavy isotope of nitrogen. After several generations, all of the bacteria DNA molecule contain heavy nitrogen. DNA was extracted from a sample of the culture, mixed with cesium chloride solution, and spun at a high speed in a centrifuge. In this process, DNA molecules of different masses separate into bands at different positions in the centrifuge tube. The heavier the DNA molecules, the closer to the bottom of the centrifuge tube that a band forms. The diagram shows the position of the DNA molecule containing heavy nitrogen in the centrifuge tube. The culture of bacteria was then allowed to reproduce using nucleotide containing the light isotope of nitrogen. DNA samples were taken and separated by centrifugations after the bacteria had divided once and again after the bacteria had divided twice. In which position could the DNA be found after the cells had divided once and after the cells had divided twice? The answer will be after dividing once, all at L, and after dividing twice, the position is half at K and half at L. The diagram represents a molecule of ATP. What are the components of ATP? Label P, Q, and R. The answer will be P represents purine, Q is pentose, and R is phosphate. The table shows 3 anticodons for different amino acids, which DNA triplet on the DNA template strand codes for the amino acid. To determine which DNA triplet on the DNA template strand codes for serine, we need to find the complementary DNA triplet for the anticodon of serine. The anticodon for serine is UCA. To find the corresponding DNA triplet, we first convert the RNA anticodon to the corresponding DNA codon. The RNA anticodon UCA pair with the DNA codon AGT. Since U pair with A, C pair with the G, and A pair with the T. Therefore, the DNA triplet that codes for serine is AGT. The correct answer is not among the given options, but based on the choice, TCA is RNA sequence that matches the anticodon given. So the closest answer will be TCA. 26. The photomicrograph shows a transport tissue in the stems of a grape vine. What is the structure labeled T? T is represents sieve plate. The diagram shows transfer sections of part of a plant. Transport tissues are labeled 1 to 6, which rows show tissues that mainly transport water and tissue that mainly transport sucrose. The answer will be tissues that mainly transport water will be number 4 and 6, and tissues that mainly transport sucrose will be 2 and 3. What would increase the rates of transpiration? To increase the rate of transpiration is increasing light intensity, whereby it helps because it opens stomata, which allow more water to evaporate. And which properties of water are important for transport in xylem? For water transport in xylem, the key properties are, first is cohesion. Water molecules stick to each other, which helps water move upward through the xylem. Second, we have adhesion. Water molecules stick to the wall of the xylem vessels, aiding in the upward movement. And good solvent properties, which help in nutrient transport. These are the statements which contribute to the water transport in xylem. Which statements about the blood flow in the cardiac cycle is correct? During the phase of the cardiac cycle, blood return from the body to the right atrium via superior and inferior vena cava when the eighth atrium is in relations. So the answer for this will be C. Which of these statements about the formation of hemoglobinic acid are correct? The answer will be all. It depends on the formation of carbaminohemoglobin. So this is part of the process where carbon dioxide binds to hemoglobin. And statement number 2 is pH regulation. Hemoglobinic acid help prevent blood from becoming too acidic by binding excess hydrogen ion. And finally, we have statement 3, which is also considered is correct. We have carbonic hydroxide. This enzyme help convert carbon dioxide into carbonic acid, which affect hemoglobinic acid formation. What can combine with the Hams group of the hemoglobin molecule? We have all. We have oxygen, we have carbon dioxide, we have carbon monoxide. Oxygen, where hemoglobin binds to oxygen to transport it through the blood. Carbon dioxide, although it primarily binds to the amino group of hemoglobin, some carbon dioxide can interact with the heme group. And finally, we have carbon monoxide, where hemoglobin can also bind to carbon monoxide, but this is a much stronger bond compared to the oxygen. So as a result, the answer for these questions will be all. Which factors would help a person to adjust from living at a low altitude to living at a high altitude? The answer will be statement 2 and 3. Why statement 2 and 3? An increase in the oxygen carrying capacity of the blood. And as a result, it carries more oxygen. And number 3, an increase in the output of blood by the heart. As a result, and the functions is to deliver more oxygen to the tissue. So the answer will be 2 and 3. Chronic obstructive pulmonary disease, so-called COPD, include bronchitis and emphysema. Which symptom is specific to emphysema? Emphysema is characterized by loss of elasticity of the alveolar walls. And this loss of elasticity leads to the disruptions of alveolar walls and a reductions in the surface area available for gas exchange. The other symptoms listed are more associated with chronic bronchitis. Therefore, the answer for 34 is loss of elasticity of the alveolar wall. 35 is a common question which is also already exists in October 2021 and May 2018. And the question is related to bronchus. In this question, there is a little bit of twist, but then is all the answer is towards bronchus. The question is asked about what identify the structures present in a bronchus. So, in the bronchi, you will find cartilage. The function of cartilage is to provide structural support. Cilia is to help move mucus. And finally, you have goblet cells, which the function is to secrete mucus. So the answer for this is C. 36. Which of these terms can be used to describe the role of mosquitoes in the transmissions of malaria? The role of mosquitoes in the transmission of malaria is best described as vector. Why vectors? Mosquitoes are vectors because they carry and transmit the malaria parasite from one host to another. The malaria parasite itself and the term pathogen refer to the disease which cause organism, not the insect that transmit it. The answer for this will be vector. Bacteria that infect skin wound can become resistant to the antibiotic commonly used to kill them. Which statement explains how this bacteria could become resistant to the antibiotic? Random mutation in DNA allows some of the bacteria to survive in the presence of antibiotic. Some children are born with severe combined immunodeficiency, is so-called SCID. Children with this inherited disease do not normally have any T lymphocytes and suffer from many infectious diseases. And how many of these children can be cured from SCID? Children with SCID can be cured by treatment with stem cells. Stem cells is a therapy which can replace the missing immune cells, which help to restore their immune system. The hybridoma methods is used for the productions of monoclonal antibodies. Which two types of cells are used in this method? The hybridoma methods uses tumor cells and B lymphocytes. In this method, B lymphocytes which produce antibodies are fused with tumor cells, which can grow indefinitely to create hybrid cells that produce monoclonal antibodies and can be cultured indefinitely. And a vaccine is used to create artificial active immunity. After being given a vaccine, it will take a period of time before a person develops long-term immunity against the disease. Which statements about this period of time explain this delay? The answer will be the primary immune response has not produced enough antibody. Because it takes time for the immune system to produce enough antibody after vaccination. This delay is due to the time needed for the initial immune response to build up. That's all for today's presentation and sessions, and hope to see you again in the next sessions. And please don't forget to subscribe to Germ Study Lab YouTube channel. And I wish you all the best in your studies and good luck in your upcoming examination. And finally, thank you very much for your support and cooperation.